Okay, let's get started. Thanks everyone for coming out to BrainMap. It's a special pleasure for me to welcome Todd Constable from Yale back to the Martinos Center. Todd was my Ph.D. advisor at Yale, and so it's great to welcome him back to Martinos after about 6 years. He did a BrainMap back in 2011 or 2012, and there's a lot of work from his group that we'll get to hear about today. Todd is director of Magnetic Resonance Research Center at Yale, and his work has really been trailblazing both on the acquisition side of fMRI and on the neuroscientific applications. So I think that Todd's career has really straddled both the MR physics and applications. And today we'll hear more about the latter, focused on his work with functional connectivity. So Todd, thanks for coming up and joining us.
Great, thanks, Jason. It's great to be back here, actually, and see so many friendly faces and familiar faces that I know. I'm going to allude to my last visit later in the talk at some point too. But and Jason, thank you, and Larry and Bruce and everybody for having me here. It's great to be here. So I'm going to talk about the importance of brain state in relating functional organization to symptoms and behavior. And I think what I want to get across is a few kind of simple messages, but I think we have some data that supports that maybe these thoughts are real. So one is that the functional organization of the brain is actually relevant, and is that okay?
A little louder in the back. Okay.
The functional, can you guys hear me at the back there at all? No. Somebody wave or, oh, not really? Okay. Is there a volume? Oh, that's the light.
It's all the way up. This is the light.
That's a light.
Maybe lean into the mic.
I'll speak louder. So the functional organization of the brain is relevant, and it tells us, it turns out that it actually tells us something about the person. We can call these functional phenotypes. So if we look at the organization of each individual brain, that's a phenotype, and we can extract some information from that, hopefully. I'm going to try to argue that we can. And this may be useful for categorizing patients, selecting treatment strategies for patients and things like that, and or just for learning about brains and personalities and various traits that people have. So I'm also going to show that it's possible to build predictive models of symptoms or behavior from these functional phenotypes. And so that's really doing the brain function relationship and trying to establish that. So I'm going to describe how we measure the functional organization of the brain. I'll talk about some factors to consider when doing this, and I'll talk a little bit about what this functional organization can tell us. If you're going to go to sleep, here's the take-home messages right now, okay, up front, and I'll come back to this. The bottom line is there's a lot of information contained in the human connectome. I'm going to argue that many, traditional sort of task-based fMRI studies may be grossly oversimplified, and I'll try to present some evidence for that. I'm going to also do something provocative, which is to say that the functional organization of the brain is very fluid. I say provocative because there are people that think that it's not. But we all probably think that it is. And in particular, I'm going to say that there's no single functional atlas. So we just finished a paper recently about this. So let's get right into it. The basics of functional connectivity. Basically, in order to build a human connectome, we need a couple ingredients. We need to divide the brain into nodes. So we have some sort of brain atlas here, and we divide the brain into nodes. And we then look at the resting state bold signal. So this is the fMRI signal that we get for 20 minutes or something in the magnet. And we look at how the different brain areas are correlated, and or that correlation tells us how they're connected. So this is the whole basis of functional connectivity. And in the early days, all functional connectivity was from one region to another, or from one region to every other voxel in the brain. And now, more recently, we've been getting into this whole connectome thing where you have an atlas, and you can label the rows and the columns of the atlas by the node number. And then when you look at the green node and the red node, you go to the intersection of what is it, number 5 and number 10 or something, and that's where that connectivity number goes. And so you can build up an entire connectome, an entire matrix like this, which is your functional connectome. Okay, so the whole talk is going to be focused on this functional connectome. So there it is. This is actually more what it looks like. So that was a small sample of it. If we have a 268 node atlas, there will be 268 rows and columns here. And so the matrix looks more like this. And really, because it's symmetric, we don't have directional information in fMRI. So we don't know what's driving what. So the connections between A and B are the same as the connections between B and A. And so it's symmetric across the diagonal. So given that, this is your matrix, your functional connectivity matrix. We're really looking at 1/2 of it. But there's potentially a lot of information here. So for a 268 node atlas, and I use 268 because that's what our atlas is, that's about 35,000 connections. And for a 360 node atlas, and I'll explain why I use that number, that's about 64,000 connections. And what I'm going to argue today is that sets of these connections tell us something about you. So the premise is that how the brain is functionally organized, which is really these connections, tells us something about the individual. And our goal then is to understand the link between this functional organization and behavior. So we want to establish both normal functional organization patterns linked to a range of behaviors. And it'd be great to link this to developmental milestones. It'd also be great to link this to clinical symptoms and disorders. So I'm going to describe how we build what we call connectome-based predictive models. That's the key to linking these to behavior. I'll give you some examples where we've done this. And then we'll spend most of the time talking about factors that influence these models and also some future developments. So to build one of these connectome-based predictive modeling, what we're trying to do is take these matrices from a series of individuals and find some relationship between brain and behavior. Okay, so we can get a set of individuals, let's say subjects 1 to n, and we can get a score, let's say a behavioral trait or a behavioral performance in a task or a symptom score. And so each person, subject 1 contributes a matrix, and they contribute some sort of behavioral score or clinical score. And each subject contributes their own matrix and their own score. And together, we can then go through and we can correlate the elements in the matrix with this vector here of scores. And we can come up with edges. So the edges are those connections. We can come up with edges in the matrix that correlate either positively or negatively with that score. And so what we're doing there is we're extracting across people, we're extracting the features that tend to vary with that behavior. And we can do that. You'll get positive correlations and negative. You can treat those, we generally treat those separately, but they can both go into the same model. And what we do then is we've got those edges. We can get some summary statistic of these edges. And in the crudest form, all we do is add those up. I'm sure some of you machine learning people could do more with this than just add them up. But we add up these edges, we get a network strength measure, which is the sum of those edges, and we get an observed performance score for each individual. So each individual contributes 1 dot to this plot here, and that plot then establishes a relationship between those edge strengths and that behavior or symptom or whatever it is. We can then, fit that model, fit a line, let's say, to that. So our model is y equals mx plus b. It's not very complicated. And then we can bring in a new subject that we left out, and we can say, okay, what's their network score? And given their network score, what would be their predicted behavioral score? And we can make a plot like this. We can look at what their observed behavioral score is versus what their predicted behavioral score is, and we can do kind of a leave one out or a k-fold, whatever. Nowadays we're doing k-fold. When we first did this, it was leave one out. And so you can repeat this for different subjects, and ultimately you fill in you go through all the subjects, and you get a relationship between predicted behavior and observed behavior. And that tells you something about the quality or the goodness of fit of your model to predicting this behavior. We can do this for the negative tail as well, get another model. And so that's another model relating predicted and observed, and you can actually collapse these two together and get the same thing. So this was the basis of a couple of papers, one in Nature Neuroscience and one in Nature Protocols. And I'll pause for a minute here and tell you the last time I gave a talk here, Emily Finn was in the audience here, and she was currently working at MIT Press, and she came over for the talk, and she came to grad school afterwards, and this is what she did. But you know, so that was, the last time I was here was prior to Emily becoming a PhD student, so kind of cool. She's a postdoc now at Peter Vanatini's lab at NIH. So this was, there's a couple things that were in this paper. One is that we established that that connectivity matrix for you is unique. So if I have everybody's connectivity matrix in a pile, and I have a sample of your connectivity matrix, I can find another connectivity matrix of you from that sample. So I can identify you based on this connectivity matrix. So that's where this functional connectome fingerprinting comes from. We didn't expect, actually when we weren't looking for this, we wouldn't expect these to be unique. We thought everybody would kind of look the same, but actually they don't. So that's interesting. And then it turns out that when you do this sort of predictive modeling, it turns out that, you know, how your brain is wired, those functional connections reflect something about you, in this case, behavior. So in Emily's paper, the first paper, we looked at fluid intelligence, and I'll come to that in a second. This data was originally done on the Human Connectome Project data, where people were imaged twice. They were imaged once on day 1 and once on day 2, and those were, you know, 24 hours apart. So what we didn't know initially was, you know, do you look the same a year from now, or 2 years from now, or 3 years from now? So this paper, I'll talk about another paper here that we did, which is to get some longitudinal data. And in particular, if we focus on this data from some Pittsburgh cohort here, there's 93 subjects aged about, on average, 15 years, plus or minus 3 years, and they were scanned over a 3-year period. So we could look at the 1 to 3 year period in this data and look at our ID rates. And it turns out that your connectome, we can still identify you by your connectome, even with data that's, you know, 3 years later. And this is at a, you know, development, a rapid development stage of life as well. But we still don't really know how these change when they're established in infancy, and how they change over time or development or with aging. And so that's an area of interest that we're still looking at. In Emily's paper on the introducing this approach, we looked at fluid intelligence, and we saw that we could get some predictive value in terms of trying to predict people's fluid intelligence by sticking them in a magnet and looking at their resting state data. You can get some more information from this too. You can look at this in terms of localization. So if we take our 268 node atlas and we divide it into networks, this is dividing it into 8 canonical networks. There's kind of a sensory motor network there, number 5. Number 3, I think, is default mode network. 1 and 2 are frontal parietal networks. And then we can look at the contributions of those networks to our models. And we see that a lot of the frontal parietal cortex or connections within the frontal parietal cortex contribute quite a bit to the model. And a lot of default mode areas contribute in the negative sense. So the weaker the connectivity between some of those regions, the better you do, or the higher your fluid intelligence. But you can also see that there's basically contributions throughout the brain. And I'm going to come back to this point too, that the whole brain is kind of involved. This does generalize. I think I have a slide coming up showing multiple publications on multiple different behavioral scores and things like that. But here's in adolescence, this is from the ABIDE data set, looking at social responsiveness scores in autism. And the healthy controls are the gray colored dots here. And they're all basically have the same score on this scale. And then the autistic children are in the colored dots. And we can see there's a total SRS score that relates, or we can somewhat predict the total SRS score using an approach like this. There are subscores in autism. These are pretty substantial numbers of subjects here, and so we can do pretty rigorous k-fold testing and randomization to make sure that these results are valid. But basically here we can build models that predict these various scores. Now the model isn't everything, so we don't necessarily want to replace cognitive testing, right? What we want to do is we want to understand the source of these changes. So what are the connections that are involved in this? And what does that tell us about the neurophysiological representation of these disorders in the brain? And maybe we want to be able to, based on the functional phenotype, stratify people into certain treatment categories and things like that. And I'm going to come back to that point later. There's also an ADOS score in that data set. And so we could do the total ADOS score and subscores in that and get pretty good predictive values. I talked about randomization. So this is versus randomization. So there's a trained test and a randomized data set here. And these results are all significantly above the randomization scores. And so these are statistically reproducible results, and this is with split half permutation testing. So that was with the ASD, the SRS scores, and the ADOS scores. Again, showing that these results are robust to noise, basically. So in what I've been talking about, now I want to get into kind of the more subtle features of this. What I've been talking about so far is that each subject contributes one connectivity matrix and one behavioral score. And from that, so remember there's between 35,000 and 75,000, let's say, edges in here. And some subset of those edges contains information on, let's say, fluid intelligence, ADOS score, SRS score, things like that. We could build multiple models out of this data, and those different models would probably pull out different edges. Some of them might pull out some of the same edges, those edges being connections between two nodes. But the bottom line is that they're, given the right data, we could build many, many models of behavior from this single connectivity matrix. So that's one take-home message. There's a lot of information in this matrix. Also, the matrix doesn't necessarily have to be from resting state. So far, everything that I've talked about has been from resting state, and much of what the field is looking at is resting state data. And actually, when I got into resting state data, I thought, oh, good, I don't have to do tasks anymore. And tasks are complicated, and there's all these subtleties with various cognitive functions that you have to take into account with tasks, and it's very difficult. And so I thought, oh, great, I can do resting state, just put somebody in a magnet and image them, and that's pretty easy. It turns out that it may be that tasks can tweak or stress the brain in certain ways that are more informative, and that's what I'm going to try to show you now. So most of the data to date has been focused on resting state, and the question that we're going to address right now is resting state the ideal condition to collect connectivity data? So brain state manipulations, our premise is going to be that brain state manipulations actually can reveal traits. Remember, the title of this talk is The Importance of Brain State in Relating Functional Organization to Symptoms or Behaviors. So I'm going to show you an attention study now that we did. So this is where we had both resting state data and an attention task called the grad CPT task. It's a continuous performance task with pictures that gradually change from one frame to the next very rapidly. And you push a button every time it's a city scene, and every now and then a mountain scene comes up, and you have to withhold your button press. So you have to pay attention. It's fairly easy if you do it for 10 seconds. It's actually really hard if you do it for 6 minutes. So it's a continuous performance task. You can perform in the magnet. You're doing the task the entire time. And then we treat the data just like resting state data. So you still get a 6-minute block of brain volumes. And in one case, you've got a 6-minute block of brain volumes collected during rest. In another case, you've got it with grad CPT. Plus, we get in-magnet performance data. And so we can use that to measure how good the attention of that individual is. So we did this on 25 Yale undergraduates. We built the attention connectome-based predictive model on these undergraduates. And the cool thing about this work is that we were then able to take this model. So then we had a model of attention. So what edges are strong and what edges are weak in your brain if you have good or bad attention. And so we can build a model that would reflect that, and we could then take that model and apply it to some data from children that were imaged in China. And these children had ADHD scores, which is a measure of attention. Maybe not the same measure of attention that we use to generate the model, but it's still a measure of attention. So in building the model, we can do a leave one out analysis and look at how well our model does in the Yale group. And so that's what we have here. So this is predicting the in-magnet D prime behavior in this sort of leave one out framework with these 25 subjects. Now there's a couple of things to, you might take this with a grain of salt because A, it's a very small sample size, 25 subjects. We have in-magnet performance, and there's a, severe potential here for overfitting. We have amazing numbers here in terms of the predicted versus observed D prime, but I'm just going to leave that as it is for now because the cool thing is the generalization of this model to another population. So we can also then take that model and apply it to the resting state data in the same group of subjects that we collected. And we see here that the fits aren't quite as good. So the predicted versus observed fit isn't quite as good, meaning that the model isn't performing as well when you're looking at resting state data. So that would say it's something between, there's more information, or the edges are more revealing, the functional connections are more revealing when the data is taken during this sort of attention task than when the data is taken at rest. And these were interleaved, by the way, so there's no, there were multiple runs, they were interleaved, so there's no run order effect here or something like that. So we have these two data sets. We have connectomes from resting state and grad CPT data. We can then take this model and apply it to, there were 113 kids aged 8 to 16, imaged in China, different imager, different pulse sequence parameters. I think they didn't have multi-band, so it was long TR, things like that. Apply it to the resting state scans and try to see if we could predict the ADHD scores of these subjects using this model built in Yale grad students. And it turns out we could actually do pretty well. The model goes in the opposite direction here because ADHD, a high ADHD score is like low attention, right? So these are opposite measures. But so here we have like an R of 0.3 or 0.34 or something is actually pretty good for this sort of thing. And so this says that we can build this model in the Yale students and get some predictive power in terms of predicting ADHD scores from in another population. So that was pretty exciting, actually. We could turn it around and we could try to build a model in the ADHD kids. And the advantage here is that potentially we have more subjects here. We have 113 subjects here versus 25. So maybe we can build a better model going the other way. And noting too that this is remarkable success given that these are different scores, different measures of attention that are building these models and testing the models. So if we turn it around and we build a model on the using the kids in China and apply it to the Yale students, we still have pretty good predictive. In fact, it works better going in the reverse direction. So we're able to predict the network strength from the task and from the resting state data. So the top row is the task data and the bottom row is the resting state data. So this was pretty cool. I mean, it brings up a few things. One is that task seems to matter. Also, the number of subjects seems to matter. So we can build a better model coming back this way than going forward. But it was remarkable that we were able to build this attention model on one group and test it on another, and that it worked in such different groups. So this was a Nature Neuro paper. Monica Rosenberg was a grad student in psychology with Marvin Chun at Yale. So that was encouraging. We then, with Emily, went on to look more at brain states. So what are the brain states and how do brain states influence the edges? And I think of it as, in the original fingerprinting paper that Emily wrote, we showed we were using the HCP data, and in the HCP data, they have a lot of task data. That's block design task, but whatever, it's task data. And we showed that in the fingerprinting, so if we're trying to ID you, our ID rates were very similar whether you were using your resting state data or using task data. They had motor tasks and language tasks and memory tasks and things like that. And our ID rates were very similar, which means that you can, my interpretation of that is that you can tweak the connections a little bit with a task, much like when you do task-based fMRI, you get a 2 to 4%, let's say if you're lucky, signal change, and so you tweak the system a little, but for the most part, the brain is as it is, and that's why we can ID you still, whether you're doing a task or rest or whatever. But we can tweak it potentially in meaningful ways such that we can get more information from it or build better models, and that's what we're investigating here. So I think of a task then as analogous kind of to a cardiac stress test. So if you have everybody at rest, everybody, if we looked at your hearts now, everybody's probably got a decent resting heart rate and things like that. But where we would really see differences in heart function is if we made everybody exercise for 20 minutes or something like that. So I think this is where the brain is, when you make the brain do a task, you start to pull out differences in capabilities and or intrinsic functional organization. So the question is, what do we use for that cardiac stress test here? So what task? So a set of tasks may best characterize any particular cognitive function. I'm going to come at this from the physicist point of view, which is I don't know which task is right. There's probably no right task, but maybe there's a space that we can span such that we can find, from that space, we can find some orthogonal component that reflects the cognitive dimension that we're interested in. So I would propose that we should look at a range of tasks, tweak different aspects of cognitive function with those, and then extract, models from that composite data. Now, of course, it also depends on what you're trying to model, right? So if you're interested in delusions and paranoia, maybe you want to tweak those systems a little bit with movies or stories or something that are tapping those sort of systems. And so, and that may well be. So my goal here would be to have a suite of tasks that could help with this. To test some of this out, we went back and looked at the HCP data again. And so here's now in Emily's first paper was the first release of HCP, I think it was 118 subjects or something. Now we have 515 subjects in this. And we also looked at the Philadelphia neuroimaging cohort data, which is 571 subjects. And we could build predictive models. So here we're trying to predict fluid intelligence, and we could build models for all the different tasks that they had in the HCP. So they had, it's kind of hard to see here, but they had gambling, working memory, emotional memory, a motor task, a language task, a social responsiveness task, a relational memory task, and then 2 rest runs. And you see that the rest runs, as the percent variance explained, the rest runs were actually the worst for trying to build a model to predict fluid intelligence. And actually the gambling task seemed to be the best in this case. So maybe whether you gamble or not is a good measure of fluid intelligence. No, it's not whether or not, it's how your brain responds in this task. But so here we can plot then the fit between the observed and the predicted fluid intelligence, and we show here that working memory and emotional memory tasks do quite well. The reason we're plotting working memory and emotional memory here and not the gambling is that we wanted to compare with the Philadelphia neuroimaging cohort, which actually showed the same thing. So there they had sort of equivalent working memory, emotional memory, and rest runs, and we get the same sort of response in building these models in that data as well. So this would suggest that it is possible to tweak the brain by doing tasks in a manner that would allow you to build better models when you're doing this sort of connectome-based predictive modeling. So this was a paper we had in Nature Communications just recently. So task-induced state manipulations improves the prediction of individual traits. Now, one of the problems with all this publicly available data is that it wasn't designed for this purpose. And so both in the Philadelphia neuroimaging cohort and in the HCP data, those were block design studies. And really, what we showed in the attention task that I showed you earlier, that's a continuous performance task. And I think continuous performance tasks are probably cleaner in the sense that your brain is getting into this state while you're doing this continuous performance task, and that helps us to build those models rather than oscillating between states when you have rest or some control condition versus a task condition. So we have since collected some continuous performance tasks, not of the order of 500 subjects, but I'll show you some of that data in a minute. So the attention data would suggest that these continuous performance tasks should be better. So what we should be doing is that we should be looking at a series of continuous performance tasks. We then need to do modeling to build these all together, tweak the brain along these different dimensions, and then we can potentially extract even more information from it and account for more of the variance when we're trying to predict performance. This is well suited, and Anastasia is here, and she has a nice YouTube talk on RDoC, formalism, and building predictive models of behavior. But if you're unfamiliar with RDoC, so in psychiatry, there's a push to sort of a dimensional approach to symptoms. So rather than trying to categorize people in specific bins according to the DSM-5, the idea here is that you can look at symptoms. Everybody's on the spectrum somewhere. So we all have some level of paranoia. We all have some level of delusions. We keep submitting grants over and over, and they keep getting rejected. You know? So don't deny it. So everybody's on the spectrum, and the idea is that, you know, this spectral sort of domain is ideal for this sort of you know, connectome-based modeling where we can, you know, build a model of how your brain varies with a certain symptom or something like that. And so they've defined, in defining this RDoC, which is this research domain criteria, they've already defined for us a number of you know, cognitive constructs, for example. And so what we've done is we've picked a selection. We can't do all of these in the magnet on one person at one time, but we picked a subset, you know, looking at attention, perception, declarative memory, language, cognitive control, working memory. Those are constructs that we can take and do like a 6-minute run, and we can do all of these in sort of within a session and collect data like that. And so that's something that we're working on right now. And so that ties in nicely with this, the title of the talk again, which is this brain state manipulation. And now maybe none of these tasks is ideal for extracting information on your attention system or your perception system or your, you know, level of schizophrenia. But all these systems are involved in those systems, and maybe there are dimensions along within this space that span that, the important dimension that we're interested in. And so that's what we need to sort out still. So I'm going to go into the, so this is the more speculation, and I'll try to prompt your thinking a little bit more here, being a little more provocative. But so recall we work in the field of human brain mapping. Okay, that's what this is all about, or some of this is about that anyway. There is a field called human brain mapping, and there's a journal called Human Brain Mapping, and there's an organization called Human Brain Mapping. So when we predict behavior, we want to ask, well, you know, a neuropsychologist will come in to you and say, well, what's driving this? You know, and the answer they want is the amygdala or the hippocampus or something like that. Right? I would say that the evidence we're starting to accumulate from this, and I'll talk about some other evidence too, is that, you know, maybe that's an oversimplified view of what's driving it, and that what we find with CPM, despite many, many attempts at trying to get very localizing with this, it's not very localizing. So we see relevant circuits basically throughout the brain. And potentially this is correct, right? I mean, the brain might be more complicated than like the amygdala is driving this, and that's it, you know, that's the story. So presumably different circuits in the brain don't act alone, it's all integrated. And this is maybe, you know, for the first time getting at some of the complexity of the brain. And so I'm going to say that our tendency is to, and the reviewer's tendency, is to want us to nail this down to like, well, what are the two areas that are driving fluid intelligence or something like that? And I think the answer is going to be, it's not that simple. So if we look at, typically in these models, when, remember I said there's 35,000 edges, let's say, in the 268 node atlas. Of those 35,000 edges, we might actually get something like 200 connections that come into our model to predict fluid intelligence or attention or something like that. So 200 edges looks something like this in one of these circle plots. So we have the front of the brain here, the back of the brain here. This is the left hemisphere, this is the right hemisphere. And then these are different regions. So prefrontal moving from front basically to back here, cerebellar back here. So these are nodes then along here, there are little dots, and each of those dots represents a node in the prefrontal cortex. And then these are, what it's connected to both within this hemisphere and across hemisphere. Okay, so that's what these circle plots show. These circle plots then are showing that in the fluid intelligence model in the top row, in the attention model in the bottom row, basically we're getting connections all over the place that are involved. And so we could go back and we could say, well, this is probably highly noisy and I don't believe that. Or maybe we need to rethink this and think, well, the brain is complicated. Maybe all these things are working in synchrony, and this is actually pulling out something real. This makes it really, really difficult for us to interpret because we always want to go back, because we're in the field of human brain mapping, we always want to go back and say it's this area or that area, but we might end up having to say it's this circuit or that circuit. So I don't have the answer there, but I'm going to suggest that that's, I think, where we're going. Here's in the ABIDE data, these are the networks that we were pulling out, both for the social responsiveness scores and for the ADOS scores. And again, you can see very complicated systems. There is, if we take a step back and we look at the literature, there is support for this even from the task-based fMRI studies, okay? So if I go to Neurosynth, which is basically a database full of all of the task-based studies that everybody's ever published. If they publish Taylor coordinates and task activations, it's in the Neurosynth database. If you go to the Neurosynth database and you look at these regions, so each row here is one of our nodes. So there's two columns, so it's basically our nodes spread through two columns here. And if you go across the bottom, these are all various behavioral domains associated with that region. And you can see that for any given region, look at this node here, for any given region, there are a ton of different behavioral domains associated with that have been reported. There's some that are reported more than others, but clearly nodes are not acting just on one cognitive task, for example. So like hippocampus isn't just memory. Don't kill me. But systems are complicated, and this Neurosynth data would suggest that yes, indeed, symptoms are complicated. That's just blowing up some of the behavioral domains that are listed here. You can do the same thing in brain mapping, and actually there's data in PNAS from Peter Bannattini where he did, I should have a slide on that here, he did 10 hours of imaging somebody doing simple finger tapping, and they could basically find activation everywhere in the brain. So one of the problems with the fact that we localize to one or two regions in most of our task studies is we don't have enough sensitivity to be confident above 0.05 cluster corrected that there are other regions involved. But in fact, if you have 10 hours of data on a subject, you would see that indeed there's a lot of other brain areas involved. And I think it's all those other brain areas that we're sensitive to in this modeling with these large numbers of subjects. And so I'm going to argue that maybe we need to look at the brain in this more complex sphere to interpret some of the data. So in terms of spatial localization, these networks tend to these networks that lead to nice behavioral predictions are both extended and complex. They're not easily localized to a couple of nodes. So it's difficult to really reduce it down to that. And so I propose, well, it's not easy to comprehend, and while we always want to simplify things, perhaps it's a more meaningful representation of the complexity of the human brain. So I'll leave it at that on that point. Now, what I've showed you so far with CPM is that we have a method, this connectome-based predictive modeling, to relate the functional organization of the brain to behavior. It provides, behavior or clinical scores in the case of autism there. It reveals these complex systems that are related to this behavior. And it may, I haven't shown any data on this, but I hold out hope that, your functional profile may actually be relevant in terms of categorizing you into groups. And so that could be, but there's still much to learn about this. And so I'm super psyched about this, and some of you guys should get into this too, because there's a ton of things to learn here. But let's go back to the beginning. So remember this, I showed this slide where we have an atlas and we get two nodes and we look at the time course of those nodes in fMRI, and that's how we build the connectome, right? So I skipped over the fact that we have an atlas. And so the question is, what do we use for an atlas? And we can, the key thing is you want to identify these functional nodes. And what we mean by a node here is that some subunit that reflects the boundaries or regions within it with a uniform function. So typically when we're looking at functional data to define a functional atlas, we're looking for voxels that all share the same sort of time course. And those should, you know, be part of a functional subunit, let's say. So this is easy to do, let's say in the hand motor cortex or the primary visual cortex. You can do a task and you can isolate those, and that's pretty easy. It's much more difficult to do, though, throughout the cortex. So if we want an atlas that covers the whole brain, we need to go to other methods. And there are various, you know, many methods that have been proposed. We could just pull an atlas out of the literature, say the Brodmann Atlas. That's less than 100 nodes. They're very, very large. Turns out that you're averaging all sorts of different time courses within a given node. And so that's not very useful. Early on, there were a lot of people using this AAL atlas. It's only 90 regions. So that's a little on the coarse side too. And so too few nodes. When I started in on this, I thought, Shilin Shen, who works with me, I told her that she was going to be famous for, you know, defining and publishing the first functional atlas. So we have Brodmann Atlas, and then we would have the Shen Atlas. And so we worked on this for a while. We have a 268 node atlas, which is what I've talked about mostly. We've expanded that to a 368 node atlas. And there it's a mix of anatomically defined subcortical structures and then functionally defined cortical and cerebellar structures. These atlases are available for download. We got some papers on them. But anyway, so that's an atlas that you could use. Matt Glasser and the WashU group came out with an atlas they published in Nature in 2016 with 360 nodes, and that's why I used that number at the beginning of my talk. That's like 75,000 edges or something that you would get for a connectivity matrix. And there's a multimodal atlas, so they looked at myelin, basically looking at the ratio of T1 to T2. They have some DTI information in there, morphometry and function. And it was mostly, it's strongly driven by the functional component, but this is a very interesting problem that people are interested in. What is the ideal atlas to use? If you look at the parcellation problem in general, in the brain, there's parcellation has an H index of 113. The number of publications has been going up rapidly. If we zoom in on this in 2000 and whatever that is, 15, sorry. There's 163 papers. You know, there's a lot of papers. A lot of people are interested in this. So we came along after Shilin was working on our atlas for a long time. I started to ask, is there a correct answer? Is there a true functional atlas? And I'm going to tell you right now, the answer is no. So there isn't a functional atlas. You're not going to solve this problem and come up with the Shen atlas or the functional atlas from MGH because the node definitions change with state. So it depends what you're doing. At the resolution we're looking at here, the time courses of the nodes mix and match according to what you're doing. So we just submitted this paper here. It's under review. And with a provocative title that there is no single functional atlas, even for a single individual, that individual is me, parcellation of the human brain is state dependent. So what we did is we imaged me over and over and over. And the idea is that I would be the large sample. And the advantage of me being the large sample is that I hopefully wouldn't have anatomic variation throughout time, across sessions. And so we should have the same atlas through sessions and through tasks and everything if nodes are constant. Okay, so we collected resting state data and continuous performance tasks on those tasks that I highlighted in the RDoc section. So we had an attention task, it was the grad CPT. I watched some movies, part of The Princess Bride and things like that. I'm sick of those things. We did the NBAC task. We did a emotional eyes task, things like that. But so I was imaged many, many times. We then did these parcellations. And the node definitions should be consistent from day to day. We did this over about 8 months, actually. And the node definitions, if in fact they're fixed and there is a right atlas, they should be consistent across conditions if brain state doesn't matter. So whether I'm watching The Princess Bride, or I'm doing resting state, or I'm doing the grad CPT task, I should get the same atlas, right? And it's all me, so there's no other variation there. So what I'm going to tell you is that, yes, we did get very consistent parcellations from day to day within the condition, but in fact, we did not get consistent parcellations from day to day across conditions. And so if you put all the data in, so these are 3, 6, 8 different atlases of me, and this is, you know, they're in a they're in a plot here showing like this is the most common form, and these are the different conditions in which we collected the data in. And so movies tended to be, my brain would go in a very unique form when I was watching It's Inside Out, Up and Away or something, and Princess Bride. But so my brain would go in a certain form for watching movies, and then in slightly different forms for these other sorts of conditions. So we can look at this in different ways. This was using an exemplar-based parcellation. So we actually started with the Shen 268 node atlas, registered that to my brain, and then we had exemplars for each node. And the reason you use an exemplar-based approach is that then we always have correspondence between nodes, so we know which node goes where and things like that. And we always have a fixed number of nodes. So we didn't let the number of nodes vary. So we did this, and I'll tell you, we did this at multiple atlas sizes, and I'll talk about that in a minute. Basically, so we start with the Shen 268 node atlas, we impose it, and then we, on the data, and then we say, okay, what does it do as it finds the solution under these different conditions? And it turns out it finds different solutions consistently under these different conditions. So if we look at parcellation similarity as measured by Hamming distance in this plot here, we can see that for the diagonal within a condition is very similar to itself, but very different across conditions. And the movies is this dark line here. So the movies were very, very different. We could also do node size similarity. So we have we can calculate the size of every node in the parcellation. So the size of the nodes is highly consistent across sessions, or within a session, but very different between sessions. And here's a distributions of those measures. So you can see those distributions are very different, whether it's between the tasks or within the tasks. And so that's kind of cool. So the node sizes are changing. The center, basically the node locations aren't changing in the sense that we can keep track of which every node is. We can convert our atlas then into a node size vector. So for 268 nodes, we can make a vector of 268 points where each element is the size. And actually we could then take that vector, do a parcellation on unseen data, and say, what condition was this collected in? And so there's a predictive model, and we can actually classify that, and we can actually predict what I was doing, like which task I was doing based on my parcellation. So the parcellations change in a meaningful way between conditions and in a consistent way such that we can actually predict which condition you were in as it changes. Now, I'm going to show you this. It's a, we can also predict in magnet performance. So my performance varied on some of these tasks a little bit from day to day. And we tried to parcellate then each day separately and see if we could build a model based on performance. I don't put a lot of, this may in fact be real, so maybe your parcellation is changing according to how much you're in that state, and that reflects how much, how you perform. But this is a pretty small, it's 30 sessions. There's no test retest thing here or anything. This is leave one out with 30 sessions. But basically, this suggests that maybe we can even predict within magnet performance based on how much you're in that parcellation mode. So is this finding only true for my brain? Is there something weird about my brain? There's publicly available data from the Midnight Scan Club. They did 10 subjects, scanned 10 times, but 2 of the subjects don't have all of the tasks or all the behavioral data, so we only used 8 of them. But with 8 subjects, the data has these similar distributions, and again, within task. So they had different tasks than we did, but within task here, and these are block design tasks too, not continuous performance. But anyway, it holds, in this data with 8 subjects scanned 10 times. What we're measuring here is within a subject and then collapsing the data across subjects after we've parcellated. And so the question is, does that generalize even further? And of course, we go back to the HCP data, and we show that even in the HCP data, this holds. So there are clear differences in the parcellation you get, whether you're at rest or whether you're doing a task. So we showed that in an individual with no anatomic variation, and then we showed it in multiple individuals where even when you take anatomic variation into account, you still get this change. And so, and also we could predict tasks both in the HCP. So this is in the, this is a Midnight Scan Club, and this is the HCP data. We could predict which task they were doing based on this node size vector. So the node size vector is potentially another feature that might be of interest to this work. So what's the limit of this? If we expand, so this was with like a 268 node atlas. If we expand this to the size, if our atlas is the size of a voxel, or each node is the size of a voxel, they're invariant, right? They can't change. So at the limit of voxel sizes, your brain is invariant. Those voxels don't change. But we extended this to up to 5,000 nodes. So we actually did multiple steps. We have the 268, we did 360, we did 400, 800, 1,000, and then 5,000. And even at 5,000, we get a very similar result. So this is in my data, back to single subject data. With going to 5,000 nodes, we still have much, much stronger within condition similarity of the parcellation and differences in parcellation between. And 5,000 is about 25 voxels at a 2 millimeter resolution. That's about 25 voxels per node. So that's still, that's a factor of 10 bigger than any matrix that's used right now in predictive modeling. But they're still pretty big, so you could potentially go smaller. So here's cycling through my parcellations under different conditions. But basically, the take-home message here is that we should take into consideration when we're looking at network analysis and we're looking at changes under different conditions, we should take into, probably shouldn't use fixed nodes, and we should take into account the fact that the node definitions themselves may be changing, not just the connections between the nodes. For that, we could potentially use individualized state-dependent atlases. But the bottom line is there's no kind of functional atlas that we're going to be able to define that's analogous to the Brodmann atlas. And I'm sorry if I've crushed anybody's dreams, but that's what the data says. This is potentially a problem for brain mapping, which is, we want to take an area, we want to define it as a region and give it, some sort of rule. Maybe this is suggesting that those regions are actually quite fluid, and the functional organization of those regions isn't necessarily as stable as we think it is. Part of where, the way I interpret that, so obviously the cytoarchitecture isn't changing. Do I need to wrap up? Got a few minutes. Obviously the cytoarchitecture isn't changing, and your structure isn't changing, right? But what are we measuring with these functional subunits? I think there's probably a mix. If you think about the visual cortex, has all these cortical columns in it, right? So there's some organizational structure. There is some thought that that organizational structure does exist in the cortex as well. And I think depending on what the task is, you're potentially mixing and matching, subgroups of neurons in those regions. And whichever ones are online for the particular task, those are the ones that form a group together. And there's another set that forms a slightly different group under a slightly different condition. So this is consistent with cytoarchitecture. It's also consistent with the fact that within our blobs we have hundreds of thousands of neurons, and they're not all involved in every single task. So they're probably all mixing and matching of those systems. So that's how I think of this. So the take-home message here for my talk today is that there's much information contained in the human connectome. Brain mapping studies are maybe a little oversimplified, and I'm going to be arguing for these more comprehensive sort of approaches. And the functional organization of the brain is very fluid. There's no single functional atlas. So if we go back to this original picture here of an atlas and a connectome, so this is your functional connectome, the picture is more like this, where there's multiple atlases according to your brain state. There's multiple connectomes that have been tweaked in different ways and different dimensions, and maybe all that data needs to go into a model. And in fact, you could potentially look at this dynamically. So we did these big blocks, 6-minute blocks of condition, but your brain state is oscillating constantly. There are probably state-dependent changes through time that might be considered here as well. So that's something you guys can figure out. So thank you very much. There's a big team doing this at Yale. And I didn't really have any NIH funding for this project, but whatever. So, thanks to everybody.